I'd like to welcome all of you, in particular, of course, Professor Arturo Casadevall, to the second lecture of our this year's Robert Koch Colloquium, which is on climate change and public health. Actually, it is a great honor for me to introduce today's speaker, Professor Arturo Casadevall, who will speak on microbial virulence and climate change. I'd like to briefly introduce him. Dr. Casadevall received MD and PhD degrees from the New York University, and he's a trained infectious disease physician. He held many important positions and is currently a Bloomberg Distinguished Professor and Chair of the Harry Feinstein Department of Molecular Microbiology and Immunology at Johns Hopkins Bloomberg School of Public Health. Actually, I got to know Dr. Casadevall through his damage response framework, which I vividly teach during my time at a university professor. And to the delight of my students, they understood that pathogens don't only cause harm, but sometimes it's the host himself who is part of this. And this was a major shift of understanding in many of the students, I have to say. His major scientific interest is focused on fungi and fungi as pathogens, but much like our institute's patron, Robert Koch, he's of a cadre of scientists that tackles urgent medical and public health needs. Assessment of plasma transfer therapy against COVID-19, only the most recent example. If I list ethics in scientific conduct, biosecurity, mentoring underprivileged scientists, service to the scientific community, be it as a chair or president of learned societies, or as a member of scientific academies, including the National Academy of Science of America, I list some fields of impact of our today's speaker. Bloomberg Distinguished Professors are elected because of, I quote, their interdisciplinary leadership in bridging intellectual communities to solve critical challenges, quote end. Climate change clearly will hold critical challenges for health, and as we were reminded last week, I remember this for the whole planet. Dr. Casadevall recently advocated more engagement with this change in an article entitled Our Health, Our Action, Our Planet, a call to action for microbiologists. As a governor of the American Association of Microbiology and member of the steering committee contributed to this report of the ASM, the American Society of Microbiology, and AA Microbiology on microbes and climate change, science, people, and impacts. Dear Professor Casadevall, I'm confident to speak for the whole audience today in saying that we are very much looking forward to what you are going to share with us. We are delighted to have you, and we are actually, let me just express my gratitude for all what you have done, your scientific endeavors, and we are really, really very happy to be part of this meeting today. Now it's your turn.
Thank you, Professor Wheeler. I commend the Institute for highlighting climate change. This is the biggest challenge humanity is facing in 80,000 years. And we need to really think about it in that way. If we don't get this right, humanity has no more problems. Take a look at Venus. There is no happiness, there is no suffering in Venus. It's dead planets. As I'm about to highlight in the talk, there is plenty of past evidence about what the future will go forward if we don't address this. So I am going to, hopefully I can share content. Here we go. Is that visible? Do you see it?
Yes.
Wonderful. I feel the same. I feel we're already successful. So I'm going to talk to you about microbial virulence, mammals, and climate change. It's going to be a big picture. Hopefully something for everybody. Little on data, big on concepts, big on ideas. So we're going to talk about the microbial virulence, the concept of pathogenic potential, which is a new concept that is important to understand where we may be going, the paradoxical paucity of fungal diseases in mammals, endothermy as a protective mechanism, a fungal filter at the end of the Cretaceous select for mammals, and then the warning that global warming will bring new infectious diseases. The presentation will look at things that are very different. We're going to basically talk about mycology, climate systems, evolution, and the mammalian evolution and climate warming. My favorite area is history, and I think that we all need to learn history if we're going to for our science. And it's really, when you think about it, it is ideas and methods that are tremendously powerful. When we study mechanisms, they help us establish causality, but often it is the ideas that set the framework. And for most of European history, and actually Eurasian history, the major theory of infectious disease was miasma. And this was the idea that disease was passed by bad airs. The word malaria comes from mal area, bad airs. And this miasma theory, it began in antiquity. Vitruvius writes about it in the 1st century. I'm not going to read it to you, but you can imagine bad airs comes from the swamps and people get sick. Well, it was the Pontine marshes around Rome and the bad airs, and it was the end of summer, and it was malaria. But it's important to know that our theory doesn't have to be right to catalyze major important changes. It turns out that miasma theory led to the great public health works in Europe in the 19th century that created the aqueducts. So even though the theory wasn't right, the outcome was good. And it's led some societies to adopt cremation. Now, one of the fascinating things is, why did it take so long for miasma theory to be replaced by the germ theory? The microscope was invented in the late 1500s, but it doesn't usher a revolution, just like the telescope did in astronomy. The moment Galileo looked at Jupiter and saw 4 little dots, that upended the Ptolemaic system and began a series of changes that catalyzed most of the scientific revolution. Many things, but in microbiology, the same thing doesn't happen. And the reason is miasma theory works quite well. But in the mid-19th century, in a short period of about 30 years, these individuals, of which Robert Koch is a giant, basically made the association between microbes and disease. And that was a revolution in human history. For the first time, you could now do something about the causes. I want to point out that another German giant of the time didn't agree with this initially, although at the end of his life, he was more appreciative of the hypotheses. And I mention this because we can have disagreements. We can have disagreements in science. The good thing about science is that science is a mechanism for conflict resolution. You can do experiments, you can get additional work. I also want to point out that history is not often kind to others who contribute. And in an era where we have to make major efforts in inclusion, I want to point out other heroes that should be in stamps. Fanny Hess discovered agar. Anna Williams did the diphtheria vaccine. Marjorie Stevenson did a major contribution. One of my favorite ideas is this. They didn't know that the biochemistry in bacteria and other life forms was the same. She isolated lactic dehydrogenase, and that established the continuity of biochemistry. Very, very important idea that we take for granted. Ruth Ella Moore in the United States was the first African-American PhD in microbiology, and Jesse Price trained many and developed a vaccine against Pasteurella. Germ theory disease had the requirements. It needed a new technology that was the microscope, observation and description, and some experimentation. It did not require mechanism. Mechanism is something that we have worked in the 20th century and the 21st, and it allows us to have causality, and that causality allows you to make vaccines, drugs, and other things. What's forgotten in all this is that the fungi led the way. In fact, it was fungal diseases that were first associated with diseases. Agostino Brasi, silkworm disease, by Beauveria bassiana, which now bears his name, and David Gruby in France, a ringworm. By the late 20th century, however, we had a crisis. In the days of Koch, there were two microbes, pathogens and non-pathogens. By the 1950s, there was a problem. Organisms that had been previously thought to be non-pathogenic were now causing disease. And what had happened was medicine had made major advances and developed chemotherapy, corticosteroids, antibiotics that affected the microbiome. And the result was an altered host. And when you had an altered host, other organisms began to be pathogenic. And that led to the concept of opportunism. And the problem was that by 1990, non-pathogens have become pathogens. If you brought Robert Koch and you showed him the microbiology lab at Johns Hopkins or in Berlin Hospital, he would have been shocked that the organisms that were being recovered from patients, these would have not been pathogenic in 1900. And the reason is the hosts have been changed. And the problem is that microbe-centric theories cannot explain the changes. I just give you an example. You have a messy lexicon. Whenever you have a messy lexicon or a messy definitions, you have a problem in the field. Candida albicans can be a commensal in asymptomatic individuals, but a primary pathogen in women with vaginal candidiasis that don't have anything wrong with their immunity, but an opportunistic pathogen in AIDS. How can that be? Well, the problem is that these definitions don't take into account the host. So in the 1990s, when I was an assistant professor, Lysiane Porosky and I were asked to teach, and to teach a course in microbial pathogenesis. And we began to experiment with new ways of teaching students. And by 1999, we had new thought that we realized was new. It was new concept, having them all from teaching. And this is why I tell everyone, when you're asked to teach, teach. Because when you teach, you process information very differently. And there are numerous examples where concepts in science came out of teaching. And the idea was, how do you publish thought today? Well, thought is not greatly valued. Where would Darwin publish Origin of the Species today? He had plenty of description, but he had no mechanism. So what we had to do was hide it in mini reviews. And we have continued to do that. So what you do is you write a mini review, you review something, and then you put your ideas into it. This is a problem of our time. We value data. We don't value thinking that much. And the net result was the damage framework of microbial pathogenesis that Professor Wheeler mentioned previously. And it's a very different way to look at the problem. We don't think about pathogens, non-pathogen, commensal. We think only about microbes and hosts. You put them together and you get a new outcome. And that outcome can be, is in a continuum of damage. It can be anything from commensalism to disease. And that the basic interaction is parabolic, so that you get into trouble when you have a very weak immune response or a very strong immune response. It's very simple. It works. And microbes are now defined by the state of the host, not by the traits of what they do. So you go back to Candida. You take Candida and you put it with different types of hosts, and you get different outcomes. The organism, in a way, remains the same. It's got 5 major consequences. It shifted the base of pathogens versus non-pathogens to the outcome of the interaction. In our mind, there are no pathogens, no commensals, no opportunists, just microbes and hosts. Put them together, see what you get. And virulence is only one outcome of this interaction. You can get commensalism, symbiosis, indifference. And these states are continuous, and they differ only by the amount of damage incurred by the host. It sees virulence as an emergent property. So in 2015, we were able to add the microbiome to this, but notice that we're still putting it in mini reviews because thought continues to be difficult to publish. A couple of years ago, we were within months of COVID, we were able to incorporate all the then available knowledge for COVID into the damage framework, showing, and it basically suggests exactly what was seen in the clinical trials. Pro-inflammatory drugs work at one end of the parabola, anti-inflammatory drugs like steroids work at the other end of the parabola when people get quite sick. remarkably, we could not find a definition in the literature of what is a host. And it's actually difficult we have to think it through. Why is a rock not a host? It has a microbiome. Well, the reason that a rock is not a host and a fly, a worm, a tree, and a human are is because the above entities respond to the microbe. And when they do so, they create a new outcome, which is colonization, commensalism, latency disease. You need to have an immune system. You need to be able to respond to become a host. So a few years ago, I gave versions of the damage framework, and the Nature editor was in the audience and asked us to write about it. And what we argued is that if our words are not right, our thinking is not right. So if we go, I realize you're not going to stop using the word pathogen. It is too useful, but it is very, very important to understand its limitations. When you call a microbe a pathogen, you're giving a trait that is not its own. No microbe can be a pathogen without a susceptible host. And we worry that if the thinking was not right, people would not do the right type of experiments. And this was quite a provocative statement at the time. But I am very interested in mathematics. And one of the questions that I had was, could you develop a mathematical formalism, an equation that would help with this to put in it? And to give you a sense of what I did, I take a quote from Richard Feynman, who was a Nobel Prize winner, famous physicist. And the question is, when you hear about Einstein's equations, when you hear about Newton's equation, any equation that you know, where does it come from? Well, it turns out that it comes out of somebody's head first. People sit down and write it down, and they play with it until it works. And then they go into the natural world and they look for data to see if it works. And if it agrees with the experiment, you say, well, it's consistent with, maybe it's right. If it disagrees with the experiment, well, we call it to be wrong, abandon it. So I began playing to develop something different that will replace the idea on pathogen. And that is the idea that all microbes has a pathogenic potential. And came up with a very simple formula. The pathogenic potential is the fraction symptomatic divided by the inoculum, multiplied by an amplifier term in case there is death. If there is no death, 10 to the 0 becomes 1, and that doesn't work. So how do you use it? Well, I went into the literature and I found all those papers on the right, and I calculated the pathogenic potential for all those organisms, parasites, viruses, and all that. And what you see is it's continuous. There is no break. There is no place in which you can say this organism is not pathogenic. You can always kill a host with a large enough inoculum. And if you change the background of the mouse, you change the pathogenic potential. The formula accounts for contributions of both the microbe and the host. And it's beginning to be used, which makes me feel good, because I think that this is a different way to look at it. Every microbe has some pathogenic potential, period. Where you are and how do you use it? Well, most recently, this allows you a way to calculate the contribution of virulence factors. This is something that is relatively hard to do. If you have two virulence factor, which one is more important? If, for example, the mutant is a virulent in both cases, well, one way to do it is to calculate the pathogenic potential of both and to subtract it. And here it was done for Cryptococcus informans. So the fungi, I'm going to now focus on the fungi. They give us new lessons on the problem of virulence. The fungal kingdom is the largest kingdom. It includes the most species. They are the major pathogens of plants, insects, invertebrates, and cold-blooded vertebrates. They're currently devastating major ecosystems. The bats in North America, the amphibians throughout the world, the salamanders in Europe, the snakes in North America. But mammals are remarkably resistant. Fungal diseases don't become important till the late 20th century, when medicine affects the host. Not immunologically intact humans are remarkably resistant to the fungi. And to me, this poses the question, why? Not only are they resistant, but there are very few fungal species for humans. And they, in fact, less than 10. You cannot say that for bacteria. You cannot say that for viruses. And some of them are host associated, where you get a disease when immunity weakens in some way. and then the environmental acquired. And you tend to get them when you're immunosuppressed or one gets a very large inoculum. What are the requirements for human fungal pathogenicity? Well, you need two. One of them is it needs to be able to grow at 37 degrees or higher. And that by itself is a major problem because the majority of fungal species cannot grow at mammalian temperatures. So having that is not sufficient. Then you need to have the so-called virulence factors. You need to have the traits that allow it to survive in the face of an immune response, capsules, toxins, etc. If one has the two of this, then it has some pathogenic potential for humans. What is the evidence that endothermy protects against fungal disease? Well, if you look around the world, the organisms that are being decimated by fungi tend to not regulate their temperature. Corals, frogs, rice, salamanders, turtles, where the fungus can go right through the shell of the eggs of the turtle. When you look at human diseases, in immunologically intact humans, they all occur in the cooler parts of the body. The nail fungus, athlete's foot, dandruff, and in babies, before they have a mature immune system, you often get candida fungal rash. Experiments have been done showing the importance of temperature. So this is by John Perfect and others. And what they show is that if you take a rabbit, you cannot give it a cryptococcal infection, period, systemically, even if you put it into the brain. You can give it on the skin or in the testicles that are cooler. But if you give them steroids, then heat is not enough, and you get a dead rabbit. So what it suggests is that a remarkable resistance has two parts, temperature and adaptive immunity. And here is a very good example of this, and this is the disaster occurring in the United States today for the North American bats. A fungus was introduced probably from Europe because the European bats are resistant, and this is found in caves. It causes the white nose syndrome. And what happens is the bats are immune in the summer when their temperature is 37 degrees. But when they go into winter to survive the winter, their temperature drops to 10 to 12 degrees, and they get this white nose syndrome that is lethal. If you take the bats into the laboratory and you feed them and raise their temperature, that is sufficient to clear the fungal infection. We also showed a remarkable finding, which is that our temperature, 37 degrees, is ideally suited for controlling fungi without us having to eat all the time. So we looked at what was the temperature susceptibility of most yeasts. And we find that they all do well up to about 30 degrees centigrade. But after that, there is a rapid decline in tolerance. So Aviv Bergman took that data and a well-known formula that allows you to calculate the cost of being warm. And he asked, where is the optima? And the optima turned out to be 37 degrees. It's in Kelvin in that. This could have been coincidence, but it's totally consistent with the idea that somehow in the past we evolved, or the fungi had something to do with our evolution. And we settle with a temperature where we don't have to eat all the time, but we can exclude the majority of fungal species. And that brings us to why mammals? Why are we the dominant animals on Earth today? So the origin of mammals go back into deep time. It goes back into the Permian. The Permian, which I'm about to tell you, was one of the great epochs of life on Earth. And then about 65 million years ago, when the great calamity that wiped out the dinosaurs, the mammals were not going anywhere. They were small, rodent-like creatures living generally at night in a world that was dominated by lizards. And the question is, what in evolution will give you a need for a higher temperature? Well, there are some advantages to being warm. It gives you a stable internal environment. Your enzymes can work at one temperature. You don't have to have different rates at night and in the morning. You have the ability to move and digest food during cold weather. And I would argue, as you heard previously, that they provide tremendous protection against infectious disease. Our ancestor from the Permian would have been what is a group of animals known as the Cynodont, about 300 million years ago. So the Permian was a period that began about 300 million years ago, and it was a very, very different world. Obviously, you can see the continents were in very different parts. The flora was very different than anything that survived. And the Permian extinction, which happened about 255 million years, is the greatest event extinction in the geologic history of life on Earth. 90% of the species were wiped out. The current understanding, though, is that it occurred over 80,000 years. And this is really important because think what I'm about to tell you happened over 80,000 years, whereas what we are doing to the atmosphere is happening over a few decades. And what happened was that a magma chute went through a coal beds in Siberia and set up the coal on fire. And this fire burned for tens of thousands of years and dumped enormous amounts of carbon dioxide into the atmosphere. This is a very, and it shows you, and life did survive. We had 5% of life survive, but it probably because it was a gradual change. And this is an enormous warning to our species as to what we're doing. It turns out that there is an enormous layer of fungal spores at the end of the Permian on the boundary, 1 meter layer. So whenever the Earth does poorly, the fungi do well. They degrade matter, organic matter. So and the point is that whenever there have been extinctions, you often find a layer of fungal proliferation. Some of the Permian fossils resemble modern plant pathogenic fungi, and what is it gives you the idea that these traits were available then. That is, the capacity for virulence, the capacity for pathogenicity was almost certainly there for these organisms. So, Permian thoughts: fungal blooms are associated with major extinctions. Fungal blooms reflect the conditions of the calamity. Fungi seem to thrive at the time of planetary calamity. And need to think about fungi as filters, because whatever survives had to make it through a fungal filter for the subsequent geologic epochs. And let's go back to the mammalian lifestyle. It is enormously costly. We need 3 or 4 orders of magnitude the amount of energy that a frog needs just to get through the day. Mammals were a very minor class until 65 million years ago. They simply could not compete with the reptiles. And then something happened. 65 million years ago, and this unfavorable lifestyle became dominant. To put it in perspective, if you drive around Germany today, you see 2 major types of animals. You see birds and you see animals, and they're both warm-blooded. There are reptiles, but you have to look for them. So this is the fauna of the Paleogene, the fauna of our time, and includes everything from whales to bats to us. The usual explanation is that something was created when the dinosaurs died out and it was occupied by the mammals and birds. But to me, this makes no sense. If you had reptiles also survive, how come we didn't have a second reptilian age? If there were something had to select for this very costly lifestyle. And to put it in perspective, simply, this is what happens. The Cretaceous ends, and it's the age of mammals, the birds. The crocodiles seem to have done well, perhaps because they were an amphibian environment. So the great reptilian megafauna of the Cretaceous is replaced by the mammalian megafauna of the Tertiary. And the Cretaceous world on its end, it was a warm, volcanic, and forested planet. The temperature during the Cretaceous was 10 degrees centigrade, higher than it will be at the end of the 21st century. The sea levels were enormously high. There were forests in Antarctica. And the world was this dinosaurian flora with flying reptiles. And a rock from outer space, hits where is now the Yucatan and sets up this enormous calamity. So this calamity, within hours, the world is devastated. Giant tsunamis race through the continents. In North America, the 2 tsunamis are thought to have been several kilometers tall. And then you have a massive amount of dust into the atmosphere and fires. Why do you get fires? Because a lot of Earth is kicked into low orbit, and then it decays rapidly and it re-enters into flaming meteorites. to vegetation that is now dying. There is no sun. The sun is obscured. Photosynthesis is shut down for about 6 months. This is the perfect world for a fungal compost. There is fossil evidence that when this happened, the fungi again proliferate. And think about it, what would have happened? You have survivors, but those survivors are now facing massive amounts of fungal spores. And you have the ones, small mammals, small reptiles. The argument goes that the small mammals with their internal temperatures would have been able to move and feed in this world, devastated world, whereas the reptiles would have been in greater trouble. So I proposed this about 17 years ago. And let's talk about the post-impact world. Reptilian megafauna is killed by the blast, the tsunamis, everything at the top of the food chain is devastated. Reptiles fight off fungal infections with induced fevers, but there was no sun. Reptilian eggs are susceptible to fungal diseases. They are today. Fusarium is destroying our turtles. And you can find fungal evidence that the fungi fossil evidence that the fungi used to affect dinosaur eggs. There is now some evidence that the dinosaur is susceptible to fungal diseases. This is recently reported. The dinosaur respiratory system is avian, similar to the avian. So it means that the air went through all their bones, and spores would have landed in a lot of places where it have been difficult to eradicate potentially. And there is at least one fossil evidence that appears to be most compatible with Aspergillus, although we cannot rule out that it was a bacterial infection. There is no way to know what caused disease 70 million years ago. The post-impact world, as temperatures drop, ectotherms cannot move to digest their food. The cold-blooded animals need temperature to be able to move and find food and digest it. Temperature changes have profound effects on reptilian sex ratios. So the survivors now have eggs, but the eggs are going to come out at ratios that are not the best ratio for them to reproduce. That's going to put another problem on reptilian reproduction. And the small mammals, however, would have been able to move and forage due to their endothermy. So my proposal was that a fungal filter at the end of the Cretaceous selects for endothermy and the mammals. And this is proposed 17 years ago. It's been updated every couple of years. But the idea basically is that this very unfavorable and costly lifestyle was selected by an event. And that event was a fungal filter. And that explains why we're so resistant to fungal diseases, because we are here as a result of the fungi. So it's a very simple, by the way, I had to give it a name. In science, you got to give everything a name. Otherwise, it doesn't go anywhere. So it's been called FIMS, fungal infection, mammalian selection. It's very simple. You had two types of survivors in a devastated world. One did better than the other because they had an internal heat engine. And therefore, when the clouds cleared, when the planet recovered, there were a lot more mammals than were reptiles. And these become the founder populations that then eventually give us everything from bats to whales to humans. And it was updated recently, and plus pathogens. So FIMS has a lot of explanatory potential. It's internally consistent. We have found no obvious contradictions, and it gets updated as new data shows up. It provides a selection mechanism for an energetically costly lifestyle. It explains why most mammalian temperatures are in the high 30s. It explains the tremendous resistance of mammals to fungal disease, and it poses endothermy as a major host defense mechanism. It also explains the minor role for fever as a protective mechanism in modernity. You get a disease today, an infectious disease, the first thing doctors give you is an antipyretic medication. Why? Because today, the majority of pathogenic organisms that we encounter are already temperature adapted to mammals. If you get a virus, it's temperature adapted to mammals. You're getting it from another person. If you go into a hospital and you get a nosocomial infection, those tend to be from organisms that are adapted to humans. So it's very hard to show endothermy with the majority of today's infectious diseases. But go for a hiking trip in Germany, fall, and get a wound and get some soil in the wound. Well, you get a fever. Those organisms are not temperature adapted yet. Global warming hasn't quite hit yet. and then your fever provides a tremendous amount of protection. So the world is getting warmer, and the question is, where are we going and what's going to happen? So let's talk about the fungi, although I think that the similar arguments will apply to bacteria and apply to viruses. So today, most fungal diseases are concentrated near the equator, probably because near the equator, one has both the temperature and adaptability for them. The more of them are adapted, and the world is getting warmer. Fungi can be rapidly trained to exist at higher temperatures. This can be done in the laboratory. So I argue that global warming will bring new fungal diseases for mammals, and Candida auris, which may be the first example of this. Candida auris emerged simultaneously in 3 continents: South Africa, Venezuela, and India. And it was not known to medicine previously. And these isolates are not clonally related. So what you basically have is that you have something selected and they emerge. And this organism has come out resistant to drug already and very difficult to treat. So the argument is that this may be the first example of an organism breaking through our thermal barriers to cause disease. It was recently recovered in the Andaman Islands. And it turns out in the wild, and it turns out that the wild organisms in an area, in a marsh, were not as temperature resistant as some of the recovered from a beach where people use to frequent. So there is already evidence that the fungi are adapting to higher temperature. This is noisy data, as would be expected. It's from a European collection of yeast. And if you plot the year versus the maximal temperature, you can see that the Basidiomyces are trending up from the late 1980s. The Ascomyces serve as a control. They are already temperature more tolerant, and they are not changing. To add to the problem, humans are getting colder. So it turns out that in our world, as we get rid of many inflammatory diseases, the temperature is dropping. And I think this is going to lead to additional problems, because even a small drop in temperature combined with an increase in temperature from the fungi could bring us new diseases. So at end, I want to point out that mammals make no sense without the fungi. The thermal barrier that protects us is narrowing as the world gets warmer. The fungi are adapting to higher temperatures. I think this is trouble ahead. When you think about global warming, don't focus on the average temperatures. You got to focus on the number of very hot days. Each hot day is a selection event. New fungal diseases are predicted to emerge. Candida auris may have been the first, the proverbial canary in the coal mine. I think the same argument applies to viruses, bacteria, and parasites. So how should we prepare? We need to sample more the natural world. We need to know what's out there. We just simply don't know. Microbiology and immunology need to do more studies as a function of temperature. Most of the science, immunology, is done at 37 degrees. Your hands are much colder. We need to understand how temperature affects immune responses and organisms. But also, let's look at it optimistically. The ability to live at higher temperature could be targeted in drug development. The ability to adapt to temperature, drugs could do so and could make the organisms not viable at our temperatures. And that could be a mechanism of drug development. So with that, I'd like to end, and I want to thank everyone for this, listening to me, and I'm going to stop sharing. Happy to have made it time. To take some questions.
Great. What a fantastic presentation. Yeah, so we can start the Q&A right now. So please, if you have any question, please post them in the chat. So while we wait, I'm actually curious, coming a bit from an AMR background. I think the key discussions right now are interestingly around antibiotics and not so much antifungal. So I wonder, have they not seen your talk? So it seems that fungal pathogens.
Magda, there are limited resources and there are big problems. And, there are very few antifungal drugs. and there are very few in development. And we need to think about everything, because one of the problems is that we're constantly being surprised. But to me, antimicrobial resistance is a tremendous problem. I'm an infectious disease doctor. Infectious disease field is the only field of medicine where things get worse. I've gotten worse in my lifetime. That is, I have fewer drugs today than I did when I trained. And drugs lose efficacy. That doesn't happen in cardiology. That doesn't happen in diabetes. Insulin works just as well today as it did 80 years ago. So, but I think that we have major problems in drug development, as you know, and we need to focus on all kinds of organisms.
That's right. So, question from Wieler. So, would you then associate specific diseases of humans to the temperature they live in, apart from vector-borne diseases? Do you have any examples?
Well, the only examples that we have are really the fact that the geographic locations of many of the fungal diseases, many of them are centered around the equator. And the idea there is that the organisms that live near the equator are more likely to be already adapted to higher temperatures. So, and if you need virulence factors, it's a probability event that you're more likely to encounter them. There are very few fungal diseases in the temperate areas as you move away from the equators. So the expectation is that with global change, that is going to change as two mechanisms. One of them is geographic changes of species. And the second thing, what I'm more worried about, is that some organisms simply adapt and defeat our thermal barrier.
Hmm. So a bit connected to that with the adaptation. So I was actually surprised to hear that the human body temperature is decreasing. So
that's a really interesting observation. So what they did is, and it shows you that you can get really important insights by doing what we call low-tech research. What they did is they collected all the records that they could get from hospitals and physicals and all that for over a century. And they took and they look at the temperatures. And temperature, you can't argue a thermometer 100 years ago was not as good as a thermometer today. Temperature is something that we have been able to measure fairly well for several hundred years. So it's not a technical issue. And the reason is that in the year 1900, for example, there were a lot more inflammatory diseases. There were more people walking around with latent tuberculosis, more people having parasites in their gut. There were and it was a much less cleaner environment. So inflammation drove up body temperature, and this is what is happening now. Our temperature, Magda, is not. We are not all 37. There are some of us who are cooler, and some of us that are warmer. So if we move even a fraction of a degree, some significant fraction of the human population is going to move into the range where they are a greater threat from some of these fungi.
Oh, wow. Okay. So there's one question. Oh, hold on. Now I get all sorts of questions. Let me.
Magda has the hardest job, which is to read the questions.
Yeah.
Give them to me.
And it's moving. So
it's the hardest job of the Zoom presentation here.
Okay, so from the audience, from your perspective, beyond antifungal drug development, would you prioritize for human adaptation to climate change? Oh, sorry, what would you prioritize from human adaptation to climate change?
I think that we are an exceedingly adaptable species, and I think that we will do what we can to survive. I am an optimist. I don't think that we are going to end up with Venus in this planet. I think that humanity will respond on time. I do think that in terms of us, we cannot adapt rapidly. Look what happened at the end of the Permian. That took 80,000 years, and they still lost 95% of the species. But I do think that in terms of our own adaptation, I think that we're going to try to do what we can to cool the planet off and develop drugs to deal with these organisms and to try to figure out how to live in cooler environments. But this is the biggest challenge that we're facing. If we don't get this right, we don't have any more problems. We don't have any political problems. We don't any of the other issues. So, and I think it's important to think that way.
So, okay, so another question. So there are further differences in the immune system between mammals and reptiles than temperature. So there are different ones. Do you believe they can contribute to the selection of mammals as, oops, hold on, now I lost the, so do you believe that they may contribute to the selection of mammals as well?
Yes, I mean, I think that I like to, if you look in detail, there are major differences. There are differences between mammals. There are differences between humans and mice. But I like to look at things more globally. And when you look at backbone organisms, reptiles, they tend to have all the pieces of adaptive immunity. Frogs have really good B cells, really good T cells, really good innate immunity. They live, as you know, in water, and yet they're being devastated by a fungal disease. You take those frogs that are dying and you bring them into the laboratory, and you put them in the warm room at 37 degrees, and they're cured. So it is true that there are going to be differences, and they could have selected. But I'm looking at something global, something that had to supersede those differences. And I was always bothered by the explanation that we became the dominant group of animals because the reptiles died out. Because I grew up in Cuba and you could see iguanas. And then there had to be something else. There had to be something that selected for us. And when you compound that with the fact that our lifestyle is very costly, then it had to be a very strong positive selection. And to me, temperature becomes more important than the differences in immunity. And that the experiments with the rabbits suggest that in many ways.
Great, thanks. Another question, is there any evidence that core temperature is higher in less developed areas?
That's a good question. I don't know, is what I can say. If you allow me to speculate, I suspect the answer may be yes, if you believe the explanation that we're getting colder because of inflammatory diseases, because in many of those diseases, in many of those areas, there is a lot of chronic infectious diseases. So their inflammatory state may be higher, and that would give them a higher temperature. But this is all speculation at this time. It's worthwhile. It will be worthwhile to look into this.
Great. So that's for your next proposal, I guess. So following up on one of your last points, does it mean that areas with already now very hot temperatures could be considered hotspots of potential emergence for such adapted fungus?
Yes, and I think we need to worry about that. As you heard in the news in the last few weeks, India has gone an incredible heat wave with temperatures in some parts in the 50-degree centigrade. Think about the amount of adaptation that is going on and selection in the microbial world. Obviously we can't anticipate and we can't predict, but we need to be aware. One of the things that we are, and I ask the audience to really help with this. When you think about you listen to the discussions about global warming, listen about what is being done. There is very little talk about microbes. Microbes are the major organisms mediating elemental flows in the planet today. They need to be taken into consideration, both as consequences and as causes, since the microbes are both emitting and consuming global warming gases. So I think that we need to all become very aware and just go back a couple of weeks in the news, see what happened in India, and imagine what must be happening at the level of the environment there.
It's very true. So what time scale are we talking about?
It's scary. Again, read about the Permian. It's that is a PNAS paper. You can download it and read it freely. That was the result of coal burning over tens of thousands of years, and the net result was it set off cataclysmic changes in the planet. For us, we are doing it in a matter of decades, and I think that the good news is that we're aware of it, that we can potentially do something about it, that we are very resourceful, that new technologies can come in place to take the carbon out of the atmosphere, but we just got to get going.
Okay. So there are kind of two questions around temperature. So does the decrease of core temperature, can it influence the efficacy of our enzymes, or are they developing at the same speed?
So in theory, yes. Enzymatic reactions are very dependent on temperature. I mean, this is a well-known part of biochemistry. The changes that we're seeing are relatively small. So I don't think that we will see it in physiology unless it happens to be an enzyme that is very dependent on temperature. The majority of our enzymes tend to work over several degrees pretty well. So I don't think that a small change will manifest itself. But the second part of the question about adaptation, well, you know that you can adapt, or you can adapt organisms. So and there is a lot of redundancy in cellular physiology. So even if there was slowing of an enzyme here, you may be able to compensate with other things. I don't think that we'll see it in humans. We may see it in other organisms that have the capacity for much rapid evolution.
And the other temperature question, so body temperature in birds is higher than in mammals. Are they in general more resistant against microbes?
They are in generally very resistant to the fungi, except for one class of organisms, and that's the Aspergillus. The Aspergillus are temperature adapted. They are the major organisms you find in compost piles. You make a compost and you see smoke coming out. The center of the compost is very hot. Those organisms are temperature adapted, and those Aspergillus species can also cause disease in birds. Bird temperatures are higher than ours. They have a much higher metabolic rate, but they are susceptible to the few fungi that can grow in the mid-40s.
Wow. Okay. And do you know if there are any studies on ancient fungi species or hints of fungal diseases in extinct mammals / reptiles?
So there is some. The problem is that if you look at the fossil record, whatever disease you're looking at had to be there long enough to affect bone, because anything that killed the animal by affecting the soft tissues would be lost in geologic history. But one can find evidence of mammals, and one can find evidence of reptiles, and I show you at least one dinosaur in which the infection was long enough to affect the bone such that one could preserve it as a fossil.
Okay. If you started to collect fungi in the environment, where would you start?
I would start anywhere. The actual survey of fungi in the environment is, it's not only for the fungi, it's for the viruses, for the bacteria. We simply don't know very much. I would start in my backyard. I would start I want to know what's growing in the backyard. I want to know why I have so much trouble with my plants.
Excellent. So what would you suggest on how to assess the risk of old fungal diseases in new regions in the context of climate change? And there's an example given, like histoplasma in Europe. So wildlife health surveillance or environmental sampling?
I think both. Right now, fungal diseases are not reportable in the majority of the countries. There are very few countries that have any idea how many fungal diseases they have. France has the best system, and it still is voluntary. So we don't really know. And when you ask the question, why are they not reportable? Because historically, the majority of them are not contagious. Public health systems worry about contagious diseases. You worry about viruses. You worry about waterborne bacterial pathogens. Fungi tend to occur occasionally. So we don't even know what the global burden of fungal diseases is. It's simply not studied. And the number, and there are just probably not enough people to do a lot of these things correctly. But we have to get a better handle on this.
Are there many large-scale deep sequencing studies of different fungi species from the environment to identify new species, like lakes, soil samples, etc., much like people do with viruses and bacteria?
Yes, they are going, but the problem is one of scale. With bacterial genomes are much smaller. Viral genomes are even orders of magnitude smaller. The problem with your typical fungus is that they have very large genomes. So this is not something that is that easy to do. Even with the new technology, you can get it done. But assembly, annotation, trying to figure out where they fit is a tremendous labor. So the net result is that the genomics revolution, as is giving us taxonomy, is happening in the fungal world, but it's happening at a much slower rate than in bacteria and viruses, simply because of the size of the genomes.
Okay. So CRS is, as you said, temperature and halotolerant. What about potential to evade immunity? So how dangerous is it to public health?
So that's an excellent question. So when it first started, most of the isolates were from the cooler parts of the body. It is called Candida auris because it came from the ear. Auris is Latin for ear. However, in the past 15 years or so, now the isolates are quite thermotolerant. Candida auris has no problem growing at 42 degrees. And the majority of the cases are still happening in immunosuppressed patients. But there is great concern that it will get into the community. How is it going to get into the community? Very simple. It's going to, in places that are hospitals, it's going to get into people's skin. There is some evidence that it's already adapting to it. And then when it runs into an individual that is susceptible because of immunity, because they got a cut, because they have diabetes, then you're going to get Candida auris related disease. And that is going to be a problem because Candida auris is resistant to the major antifungal drugs already. It came out of the environment resistant. One of the things that is most troubling is that the majority of fungi in the environment are resistant to our antifungal drugs. So if something breaks through and appears, not even amphotericin or echinokindins or the azoles may be sufficient.
So, we are currently setting up several 3D organoid models to investigate pathogenic properties for infectious agents. Which fungi would be suitable candidate to be investigated in such models? And do consider these models as relevant to better understand pathogenesis in humans and the role of temperature?
First, I want to say that the organoid systems are an incredibly important tool. They will teach us a lot. Will they replicate what happens in a host? Yes and no. If you set up an organoid, for example, for a mucosal surface, you may get a much better representation than if you were studying in cell culture. However, even the organoids don't have everything in them. So what fungus will I study? I would look at what are the major problems today. Certainly, if you look by numbers, the Candida are the major problems. The Aspergillus. We're in a situation today that immunology has made enough progress that transplants are now feasible and they do well. But the biggest problem in transplants often is infectious diseases. Aspergillus continues to have it's tremendously high mortality. So understanding using organoids to look at these big problems, whether is very worthwhile. To understand how does a fungus damage an organoid? How does an organoid defend itself against a fungus? Those will be very important contributions.
Fantastic. In evolutionary terms, there seems to be a relatively clear trend to increasing autonomy of the individual from the environment, especially in the signature species. Isn't this a good enough explanation for the age of mammals and birds? Is it autonomy, fungi, or both together?
I don't know, but I have a lot of problem understanding how, if we have reptiles today and reptiles don't have to eat very much, we ended up with mammals being the dominant species. If we were crocodiles, we would only have to eat once a week. I already had 2 meals, and when this thing ends, I'm going to go eat more. Think about how costly this lifestyle is. To me, this could not have happened by accident. That something had to select for it, and therefore I go back to the question, why mammals? For birds, there may be more of an explanation. Birds need to have a high metabolic rate to fly. So maybe they were developing high metabolic rates so that they could conquer the air. And then the protection came already from that. But I do think that temperature, it had to have been selected for.
Right. So another question, this brings us back to your formula of pathogen. What is the progress beyond Paracelsus saying, can we generate impact from it?
Well, what is the progress? Originally, it was derived to try to see whether one could have a mathematical view to at least say that there is continuity in nature. We have a paper hopefully will be accepted. It was favorably reviewed, in which we have begun to look at things that the formula can teach us. So remember, how a formula, you're playing with it. What can it teach you? So one of the things we've done is we took a very simple host, Galeria, and we have been studying mortality as a function of inoculum. And we basically asked the question, is it linear? Is it exponential? Do different microbes behave the same way? And the answer is remarkable. New things have emerged in that the dependence on pathogenic potential on inoculum is different among different organisms. In fact, for some of them, it goes up. For some of them, it goes down remarkably. So the formula now opens up a whole new areas of study, because you need to be able to explain the curves. And it may be that for some organisms, the mass of organisms, the inoculum, they affect each other. They cooperate in different ways. That it's not simply a simple microbe adding to the problem for the host. Maybe there are quorum sensing effects. But that's an example where when you write something down and you begin playing with the numbers themselves generate new scientific problems that were not apparent before one had the formula.
Great. And I have a comment here that that there was a podcast featured from the Radio Lab podcast in September 2020 called Fungus Among Us. Ah, okay. I see the play with words.
In the English words, yeah.
Yeah. Well, which obviously was featured by you. And so the comment is it was very engaging and memorable. So if people are interested in sharing info on this topic with non-professional, so it's a recommendation to use these podcasts. So I think.
Thank you for mentioning it. Yeah, you can find it online. All you have to do is Radio Lab. You can put my name and it comes up right away. And I think as a scientist, the greatest honor that one could have is when the general public becomes interested in what one is doing. And I think it's very important for us to engage with the general public And the person who did Radiolab did a much better job than I can ever do in a lecture on making the story interesting. I'm just honored and thrilled to have been that this idea, because it's really just an idea. It's an idea that comes out of a brain, and then it gets filled up with data, and then it begins to generate other ideas, has been in some ways embraced and interested by the public.
Great. So I don't see, thank you. So in general, all these questions were always with thank yous, and how everybody really enjoyed your talk. So while I don't see any questions right now, but I wait if there are some burning questions. And let me tell you, I once actually saw you live at the La Jolla Institute. It must have been in 2005 when I first heard about all your theories with the dinosaurs. Obviously, I don't remember all the details, but I did have this memory of you. And I'm very happy to see that you continued along this line and actually give a wonderful presentation and connect some dots and also portray another theory. So I hope that we've got some new ideas about climate change and the impact on public health.
Well, thank you, Magda. The one thing that I would say to the participants that are still, don't be afraid to think. In science, we are rewarded by data. When you think, people say, where is the data? But what I would say to you that it is possible to think. Concepts emerge from thought. And don't be afraid. Even if they're wrong, making them wrong will promote additional thinking. Thinking doesn't cost anything. You don't need any grants support for thinking. And oftentimes, if you have an idea, the best thing to do is to start writing it down, to finding whether you can support it. And then once you are relatively sure of it, don't be afraid to get it out. And unfortunately, that's where the problem is, because journals don't necessarily often favor concepts and ideas. So one way is to do what we do, find a general area and review it and put your idea in there. But ideas are fun. They can push science forward. And I have one more suggestion for you. Every day I try to read outside my field. The reason that we were able to talk about a little bit of history, the Permian, the fungi, you name it, is because little facts were taken from many different parts of science. The way that we live our careers today, we have to specialize, and we become so specialized that it's hard to think outside of it. But it's not hard to do. I make it a point every day to read something about astronomy, to read something about history, and then and take notes. And what you do is, if you do that every day, you will get a whole set of facts that then will lead a lot of additional thought. Whatever field you're in, make connections. And I will assure you that a scientific life is a lot more fun that way than just fighting to get a paper published. Thank you.
What wonderful words can I say? I don't know if you see all the clapping and hearts. And
I see the clapping. I see the clapping.
Excellent.
If I was in Germany, I'd be hearing the knock. Thank you.
I think
we need to have a knock in the.
That's right. I think it resonates well with a lot of people who are listening, and also here at the Robert Koch Institute. Of course, I have to say that this is a lot of food for thought. I actually have one question that just came in, I guess, based on your last sentence. So is this actually a weakness of our scientific system? In physics, philosophy and theory were quite common. Should we have theoretical public health?
So we could talk quite a bit. So I am a believer that the answer is yes. Look, we become very specialized because the problems that we're working on are increasingly difficult. And the only way to make progress is to know more, to go deeper into it. The problem is that in our training, we no longer train scientists as generalists. We don't. I mean, if you think about the training of some of the giants in German science, they train broadly first. Think about, read the biography of Einstein. You know, he knew Kant. He knew the issue, some of the major philosophical problems. And then some of that reasoning was applied to physics. Today, we basically, from the very beginning, try to steer people into their areas. And then once they are in an area, we punish them if they try to get out. Well, they weren't serious if they're they weren't really serious microbiologists if they do in immunology or vice versa. We need to encourage more generalism. We need to encourage scientists to venture out. The problems that we are facing as a species, climate change, food source collapse, degradation, are multidisciplinary. They cannot be addressed with a single thing. And therefore, a scientific teaching establishment needs to begin to figure out how to restore, you know, some of these, the what we lost, what we took out of the curriculum. Here at Hopkins, and if you're interested, and if you're in front of a computer, you can look it up. We began a new program called the R3 program. If you Google or whatever your browser is, R3 and Hopkins, it comes up. We are trying to take our graduate students and teach them as generalists, while at the same time not diminishing what is so important, which is they have to do a thesis, they have to get first author papers, et cetera, et cetera. But I think that thank you for that question, because it is something that is very close to my heart and to what I believe we need to do.
Great. So I have actually a bit of a follow-up question to that. So do you think that to tackle or the forums and platforms and everything we have currently, including our education system, do you think it's actually sufficient that we will succeed in tackling climate change? Because there are always, of course, a lot of talks about transdisciplinary, or what would be your suggestion? What do we need to actually succeed?
Well, I think we need to invest in the obviously in the root causes. We need to decrease, as is being done, the putting out fossil carbon into the atmosphere. But I also think new technologies can be developed to take some of the existing carbon out of the atmosphere, and they are in their infancy today. These technologies are no different than the very early electronics of the 19th century. As they get developed, we may be able to. And let me give you a future vision. I think we're going to succeed. And I think in a future, then we take the carbon out, the arguments are going to be how much carbon to take out. So today countries fight over how much carbon you're putting out and what you're contributing. But as we begin to take carbon out, again, there will be winners and losers. Some parts will get colder, and etc. But I think that it's a much better fight, because at least we will be fighting from something that we can do. That's my vision for the late 21st century. That the arguments are going to be, where do you set the carbon limit?
Let's hope you are right on that one. Great. So I don't see any other questions in the chat. So I can only say
thank you, Magda, and you did a great job with the questions. And thank you so much for having me.
So the pleasure is, I think, more on our side. This was a wonderful presentation and all your thoughts, and also your take-home message. So thank you very much.
Bye-bye, and have a good evening.
Have a nice afternoon. Bye.
Bye-bye.